Hello students, welcome back to biology class. I'm Pratima Patil from APPA Public School. Today we are going to start next lesson, that is the fourth lesson in biology of class 8, that is cell structure and functions. Right? We are now starting today fourth lesson in biology of class 8, that is cell structure and its functions. Right? So before starting this cell lesson, you should know some of the like introduction, a basic part of the cell, where you heard actually about the cell in your previous classes, right? Starting from class 6, starting from class about this cell, right? Where you have studied, you remember in class 6, you have studied especially characteristics of living and non-living. Do you remember, students? Living things and non-living things. In this, while studying about the different characteristics of living things and non-living things, right, you have came across with a word called as cell, right? So where actually you have studied cell in this living and non-living things characteristics. So just recall of that. Okay, I think you know that the different characteristics of living organisms and non-living organisms, that is living organisms can reproduce, right? Living organisms can grow, right? Living organisms uptake nutrition, right? Living organisms will breathe and respire. Okay, and at the last, you have studied even one more point under living things or living organisms characteristics, that is, living organisms are made up of cells, right? You came across the word called cell over here in the characteristics of living organisms. That is, living organisms are made up of cells. You have studied, right? Lesson we are studying each and everything about these cells only. Getting? I have explained you only the characteristics of living things over here because I want you to recall this special point. That is, living organisms are made up of cells. Now we will study what is this cell actually and what are the different types of cells, right? What is the size of that? What are the numbers of that? Okay. Yes, students. Coming to the introduction part. Coming to the introduction part of cells, right? Who invented these cells or who observed the cells at the first time? Scientist named as Robert Hooke in the year 1665, observed at the first time, for the first time, the cells of the cork. You know, he has observed the cork cells. What is meant by the word cork? You all know the tree, right? I think you know the different parts of that. During over this part, upon the outer layer of this stem part of the tree, you'll find a brown colored hard layer, right? We even used to call it as a bark. Getting? So these cells are called as a bark cells or cork cells. He has isolated those cork cells, right? He took that a small piece of that bark or cork of this tree, made the thin pieces of that, and those thin pieces only he has placed upon the slide and observed that under magnifying glass. Under magnifying glass. Already you people came to know that the organisms which are very minute, the thing which is very small in size, which we used to see only by using either the magnifying glasses or else by using the microscope, right? by using the microscope. In the second chapter of biology, we have studied so many varieties of the microorganisms, and in that especially in the characteristics of the microorganisms, I said they observed by using either microscope or by using the magnifying glasses, which is going to magnify the image, magnify the object, right? So he has used, Robert Hooke has used this magnifying glass to observe the cork cells, right? He took the bark of the tree, made it into thin pieces, and those thin pieces he has placed upon the slide, right? And observe that slide under microscope. So what is the observation of it? What he has observed under microscope? He has observed a kind of honeycomb-like structures. Usually microscopic diagrams we used to draw in the circle, because we used to see that microscopic view or image under the eyepiece which is having a circular lens, right? So whatever the microscopic diagrams are there, which we have to draw under a circle. Inside this circle we have to draw. So what he has observed over there? He has observed a honeycomb-like structures. You can see it's a honeycomb-like structure having 6 faces, hexagon type, right? Yes, students? Right? He has observed a hexagonal type or its honeycomb-like structure under the microscope when he observed these cork cells under microscope. Right? He was like, so completely like, what is this compartment-like structure? Because at that time, no one knows that living things are made up of cells. First thing, the cork cells, and of course these are non-living one. But at the beginning, when that plant starts growing, okay, when that starts germination and undergoes different developmental stages, that time whatever the stem was there, the plant stem was there, that is completely having all the living cells only in it. But when it has grown older and older, when the barks have developed, when the cork have developed outside the stem, that time those cork cells are non-living, right? Those are non-living cells. But he has observed like this compartment type of cells, right? And first of all, he has named them as compartments only. Compartments where we used to use this word while telling the bogie numbers, compartments and all in the railway. In the train, we used to tell that, use the word compartments, right? In which compartment your seat is, right? Like that. Okay, so these are also like same compartments are there, right? And each compartment is separated with the first compartment is having one thin wall in between, so that it is going to get separate with the second compartment, right? So he has named them first of all as compartments only, because at that time there was no availability of microscope, or invention of microscope was not yet done. So after the observation of these cork cells under only magnifying glasses, right, after around 150 years, after around 150 years, when the microscope has invented, right, that time again the scientists have started observing the different cells under different type of the object under this microscope. They have used many varieties of the like objects over there, especially the onion peels, onion peel cells, cheek cells. I'll tell you what are these two later. So when they have observed, they came to know that all living organisms are made up of cells, right? All the living organism, it may be unicellular or it may be multicellular. Maybe one single cell is involved in the construction of that particular organism, or many cells are involved in the construction or the building of that particular organism. But the thing is that they all are made up of cells as living things. That's why in the beginning of the introduction only I said you the living things characteristics. What was the characteristics over there? Living things are made up of cells, right? So here, when they have started observing these onion field cells and all in the microscope, how was the observation at that time? So before that, you should know a little bit about the microscope. Okay, then I'll show how to do these onion peel cells or how to observe the onion peel cells under microscope also I'll tell you. Okay, first of all, just have a glance upon this working of microscope. Okay, how we have to handle the microscope. So this is the eyepiece, right? Which is having the handle or arm. This is the platform. Right? Okay, usually microscopes, yeah, here it is a mirror, right? So this is the eyepiece. Getting students, this is a just a rough diagram of a microscope. Observe the different parts. So if at all, if I tell you in the further classes just to observe or any onion peel cells under the microscope, then you should be able to handle the microscope. You should have like able to adjust the microscope. Okay, and here you can even see the knobs will be there. Okay, this is arm through which we are going to handle the like microscope. This is the platform where we are going to keep our slide with the object. Okay, this is the knob, adjusting knobs. So to observe the clear picture of the object, we need to adjust these rotating knobs in the microscope. Okay, and this is the mirror, right? So usually these microscope requires the natural source of light. By when during the daytime only we can observe this or use this kind of a microscope, because this image adjusted in such a way that the natural whatever the radiation of sun is falling upon this, we have to make the mirror such like adjustment in such a way that the rays should pass from these lenses, so that we can see the picture of that. This is our eye through which we are going to observe the image, right? So by using the rotating knobs, adjusting knobs, we have to make this eyepiece up and down until and unless we are going to view the clear picture of the object. Okay, and we have to adjust this mirror also so that brightness will increase inside that, right? So this is actually microscope. Why I said about this is because if I tell you now about the onion peels as how we have to observe under microscope, then you should know that how to handle that. So now coming to the first one, that is onion peel cells, how to observe or how to see these onion peel cells under microscope. So for that purpose, now what is meant by onion peel? All of you know about the onion. Right? Onion. When you cut this onion, okay, inside these layers will be there, right? Many have you have observed, right? So in the inside, in between these two layers, thin tissue paper, like wet tissue paper, how it resembles. Same like that, a kind of a thin sheet, a thin layer is available, present over there, in between the two layers of the onion, right? Open up that, okay, a thin sheet, a wet tissue paper like sheet is available, layer is available in between the two layers of the onion peels. Okay, take out that. It will be somewhat like without any particular shape. So take that one and mount it under upon the slide. Mount that upon the slide. How to do the mounting over here? Keep that onion peel upon the slide. Okay. If you go like this only viewing under microscope, we may get the like this peel cell. This peel may get dry off completely. So to avoid that, we need to add around a drop of water upon this with the help of a dropper. Right? Add a drop of water with the brush. Just make this completely stretched upon the slide. Right? Now take this slide and keep it under microscope, like upon the platform to observe the cells. adjust the rotating knobs. Okay, adjust the rotating knobs, adjust the mirror so that you'll get a bright picture, right? Adjust this fine and coarse microscope until you'll not get a clear image. Okay, when you observe clear image, what was the observation? What you will observe when you place this one under microscope? What you'll get? So, what is that microscopic diagram of the onion peel cells? It looks something like this. You are getting this picture is resembling to something else. I think you'll come to know that. I think already might have guessed about that. Yes? Yes, students, with the nucleus in it. Yes. When you take out the onion peel, mount it upon the slide, fix it with a drop of water and observe under microscope what is the observation over there. You'll find like this. Okay, when you observe that using the eyepiece, when you see that, what is the observation inside that microscope? You'll see this kind of a cell resembling as that of the a wall of a brick, right? It's somewhat resembling like as that of the a wall which is made up of a bricks. Same, right? Both are looking somewhat similar. Right? But here there is no nucleus. Okay, but looks somewhat similar. So what is that actually? Here also, whether the cells are involved to make a wall? Not like that. Actually, these are not called as cells. These are just bricks. But the arrangement, the structure of these both resembles. Both are helpful to make one particular structure. The cell and the bricks both are helpful to make one particular structure. It is making a particular building. Okay, this is making one particular maybe a organ or a cells or a group of cells which is called as a tissue, right? If we observe only about the onion cells, it is making one particular tissue over here. But other than these onion cells, if you take an example of a human being only, where many cells are going to be joined together to form one particular tissue, right? And in that tissue, all the cells are performing similar functions. That means all are doing a particular function, same at a time, right? And those tissue, many tissue which are performing, which is a group of cells which are performing a similar function, joining together to make one particular organ, right? Like this, many organs together are making one organ system in a living organism, right? So here we can say that the cells are the one which is making a particular structure, right? The cells are the one which is helpful to make one particular structure. Right. So actually, when you come to the definition of the cells, we are going to give the definition in such a way that cell is a structural and functional unit. Structural and functional unit. Why? Because we know that cells are giving a particular structure to the a particular organ or a particular organism, right? And it is performing, all the cells are performing their own function, right? Maybe different functions, but performing one or the other function. That's why we can say that all the cells are functional units also. Because of that only, the definition of cell will be cell is a structural and functional unit of living organisms. Okay, so in the same way, when you observe these cheek cells on under microscope, right? Now comes the second one, that is cheek cells. Now cheek cells, these are the cheeks, right? Cheek cells means we are not going to scrape the outer surface of the cheek. We have to take a sterilized a scraper, right? With that, scrape the inner wall of the cheek inside the mouth, right? Scrape it lightly. Okay, don't exert much pressure. It may well, cause a damage inside. Just lightly, slightly, you have to scrape it. Okay, in the scraper you'll get a purple, like a pale color, or it's a creamy white color matter in that. Okay, that one now you have to place it upon this slide. This is the matter which you have taken out or scraped from the inner wall of the cheek, right? Add a drop of water again to this with the help of a dropper, right? Spread these cells evenly upon the slide. Spread the cells with the needle evenly upon the cells, upon the slide, right? Now take this slide and observe under microscope. There also we will get kind of a cells only. With, again, cheek cells will be like this, okay, with a nucleus in it, right? So these are actually the activities which we have to do. Because of these all problems, I'm explaining right now with the help of online classes only in this way to you people. Okay, otherwise we would have conducted these practicals in our lab itself, right? So this is actually the introduction part of the cell structure and functions, right? So the next part, coming to the definition of that, coming to the definition of the cells. Yes, students. The definition of cell will be, just now I said you, if you want you can note it down. The cell is the structural and functional unit of living. Cell is the structural and functional unit of living organisms, right? I said you just now why we are calling the cell as a structural unit, right? Because it is going to give a particular structure to the living organism. It is going to give a particular shape to that living organism, right? And all the cells are performing their own function. All the cells are performing their own functions. Maybe all the cells are performing the different function, but one or the function they are performing. That's why we are calling the cell as a structural unit and functional unit of living organisms, right? So next part, whether all the living organisms, whether they are made up of only one cell or many cells. Whether all the living organisms are made up of one cells or more than one cells. We all know that because in our previous standards we have studied about that. If a living organism which is made up of only one cell, right, which is made up of only one cell, we are calling them as unicellular organisms, right? If a living organism which is made up of only one cell, then that is a unicellular organism, right? If the living organisms which are made up of more than one cells, okay, maybe millions of cells are there, then we are calling them as, yes, multicellular organisms, right? We are calling them as multicellular organisms, right? So you came to know that the presence of cells also is going to be helpful in the division of the living organisms. Okay, division of the living organisms can be done upon the basis of the presence or number of cells present in them. Okay, maybe one cell, unicellular organisms, more than one or millions of cells at a time if they are present, then they are called as multicellular organisms, right? Whether, next part, whether the shape of the cell is constant, whether the shape of the cell is constant. If you talk only with respect to the human beings only, whether inside the human body only, whether all the cells shape, no, right? There is no constant shape for the cells. We cannot say that all the cells which are present in human body are of only one shape. No, not like that. Many different shaped cells are present, right? The shape of the RBC is discoidal, okay, disc shaped, right? This is the muscle cells, spindle shaped, right? Nerve cell, I think you know about the nerve cells. In your 7th standard also you have studied in the nerve cells, right? This is the nerve cell with the nucleus in it. The shape of this nerve cell is also different, completely branched cell it is, right? Discoidal shaped cells are there in our body, such as RBC. Spindle shaped cells are there in our body, such as muscle cells. Completely branched cells are there in our body called as nerve cells, right? So we cannot say that all the cells are having the similar shape. Different shapes, right? So number of the cell may be different, shape of the cell may be different, and coming to the next part, that is size of the cell. Size of the cell. Whether size of the cell is similar in all the organisms, or all the cells are having similar size or same size, that is also not constant. We cannot say that all the cells are having same size. No, right? Cells are ranging from 0.1 to 0.5 micrometer to 170 to 130 millimeter, right? The smallest cell is having the size about 0.1 to 0.5 micrometer. What is the name of that smallest cell? That is nothing but the bacteria, right? The largest cell which is having the size of 170 and 130 millimeter. What is that? That is the ostrich egg, right? All the eggs, the hen's egg or its ostrich egg are also unicellular only, which are also made up of only one cell. That's why the ostrich egg, which is considered as the biggest cell, which is having 170 millimeter to 130 millimeter size, right? So today I have learned who invented cell, how he had, like, what at the beginning, who, like, which particular cells he has observed, that is the cork cells he has observed under magnifying glass. Okay, then how to observe, how to mount a particular onion peel cells under microscope, and different parts of the microscope you have studied, right? Next one, you have studied that how the number of the cells varies, the shape of the cell varies, and the size of the cell varies, right? Okay, students, thank you.